So we are actually talking about the lipoproteins, which are the essential part of our uh, body with respect to transportation and other physiological function. Basically, lipoproteins consist of a non-polar core and single surface layer of amphipathic lipids. This non-polar lipid core consists of many triacylglycerol and cholesterol ester molecules, which are actually surrounded by a single surface layer of amphipathic phospholipid and cholesterol molecules. They are oriented in such a position that all the polar groups faces outwards towards the aqueous medium, and the hydrophobic non-polar groups, they faces inside or away from the aqueous medium. And the protein moiety of lipoprotein is known as apolipoprotein or apoprotein. Somebody has asked earlier that what is the apoprotein? Apoprotein is just a protein moiety of lipoprotein. Okay, this is the general structure I have shown earlier in my earlier class. This actually the peripheral component of this sphere-shaped lipoprotein consists of a peripheral apoprotein. Example ApoC. There is ApoB also, and free cholesterol molecules and phospholipids. The polar group of free cholesterol and phospholipids faces outwards, and the non-polar side chains, they faces inwards towards the inwards. And the cholesterol esters and triacylglycerol, basically they are hydrophobic in nature, so they present inside the core of this lipoprotein, and basically they try to hide themselves away from the aqueous medium. So this is overall structure of our lipoprotein. How we can classify the lipoproteins? I explained it earlier that based on the density we have classified the lipoproteins into the 5 major groups. And how we calculated the density? Actually, density is calculated on the basis of their rate of sedimentation in the process of ultracentrifugation. Okay. So depending upon the flotation constant, 5 major groups of lipoproteins have been identified. And they actually play a very important role in terms of physiology and in physiological functions and clinical diagnosis. The first group includes chylomicrons. Okay, and these chylomicrons, actually, they are derived from the intestinal absorption of triacylglycerols and other lipids. The density is generally less than 0.95. Okay, while the mean diameter lies between 100 to 500 nanometer. The second class includes the very low density lipoproteins, which are derived from the liver for the export of triacylglycerol, and their density lies between 0.95 to 1.006, and the mean diameter lies between 30 to 80 nanometer. The third class includes intermediary density lipoproteins. They are derived from the catabolism of VLDL, and with the density, they have density ranging between very low density and low density lipoproteins. Like their density ranges between the 1.006 to 1.019 nanometer. And the mean diameter ranges between 25 to 50 nanometer. Actually, these all are values which one can learn, but one cannot remember this all throughout the course. For remembering these values, you should just take upper limit. Okay, like chylomicrons, they have density starting from 0.95. VLDL have density between 0.95 to 1.006, and intermediary somewhere lies between VLDL and LDL, and LDL have density like 1.019 to 1.063, and their mean diameter, we can, if we want to see, their mean diameter lies between 18 to 29 nanometer. And HDL protein involved, the HDL protein involved in the cholesterol transport and also in VLDL and chylomicron metabolism. Their density, actually they are high density, and they have their density range lies between 1.0063 to 1.21, and the mean diameter varies between 5 to 15 nanometer. Okay, and we can also remember this fact by seeing this graph, which is actually density, density versus particle diameter. The unit of density, please remember this, the unit of density is gram per ml, and particle diameter can be measured in nanometer. So chylomicrons, they are the largest in terms of diameter, and they are lesser dense as compared to VLDL. And then there occur intermediate density lipoprotein, then low density, and then high density. So one can also remember the fact by seeing this graph. Okay, lipoproteins with high lipid content will have low density, larger size, and so they float on centrifugation. So those with high protein content sediment easily and have compact size and have a high density. For example, HDL. They have compact size and have a high density. So this is all about the 5 classes of lipoproteins. Based on centrifugation, we have seen their classification, but we can also classify them on the basis of electrophoretic mobility. So lipoprotein, actually they separated according to their electrophoretic properties into alpha, pre-beta, beta, and broad beta lipoproteins. The mobility of lipoprotein is mainly depend upon the protein content. Okay, as we have earlier did that SGS page in our lab, so we know that actually protein composition of a molecule decide which protein moves faster in the gel or as compared to other counterparts. So basically, those with higher protein content will move faster towards the anode, and those with minimum protein content will have minimum mobility. So we can also classify on the basis of electrophoretic mobility. We can see this peak. There are 3 bands. This is the darker band. You can see in this image on left hand side, there are 3 bands. The darker band has higher intensity. So it is supposed to be beta lipoprotein, and the middle band is supposed to be pre-beta lipoprotein, and the lowest one is alpha lipoprotein. So you can see the concentration of these. Alpha lipoprotein is between 20 to 48, and pre-beta lipoprotein is 12 to 30, and beta lipoprotein is 45 to 70. Lipoproteins may be separated according to their electrophoretic properties, which we have seen. And now we can also classify these HDL and LDL on the basis of their protein part, on the basis of their electrophoretic mobilities. Like HDL are alpha lipoprotein, and VLDL are pre-beta lipoprotein, and LDL are beta lipoprotein, and IDL are broad beta lipoproteins. Free fatty acids and albumin complex, although not a lipoprotein, is an important lipid fraction in serum and is the fastest moving fraction too, because albumin is a total protein, so it will move faster as compared to other lipoprotein. Chylomicrons also remain at the origin since they have more lipid content, so they will remain at the starting point. VLDLs with less protein content than LDL, they move faster than LDL, and this is due to nature of apoprotein which is present. We have seen this chart earlier and we also discussed it in this class on the basis of density. These all are classified starting from 0.95 gram per ml to 1.063 to 1.21 gram per ml. And on the basis of diameter, as we go from up to down, diameter increases. HDL have lowest diameter and chylomicrons have highest diameter. Protein, if we can calculate the dry weight in terms of protein percentage, HDL have highest protein concentration as compared to others, and chylomicrons have lowest. Phospholipid concentration is also high in HDL as compared to other counterparts. Triacylglycerol percentage in dry weight is high in chylomicrons and it is lowest in HDL, which is also considered as a good cholesterol. Okay, and if we can also classify these lipoproteins on the basis of apoprotein content. Okay, so one or more apoproteins or proteins or polypeptides are present in each lipoprotein. The major apolipoproteins of HDL is alpha lipoprotein. It is designated as A. Okay, apolipoprotein A. And the main apolipoprotein of LDL is beta lipoprotein, and it is designated as apolipoprotein B, that is B100, which is also found in VLDL. Chylomicron basically contain a truncated form of ApoB. Truncated means it is half of the original size. So it is also known as ApoB, B48, and that is synthesized in the intestine, while B100 is synthesized in the liver. B100 is present in LDL, which is synthesized in liver, but B48 is synthesized in intestine. ApoE is found in VLDL, HDL, and chylomicrons remnant. So ApoE is present in all these lipoproteins. If we just consider the fact, what is the function of apoproteins in overall lipoproteins? Okay, like lipoproteins, they are the composition of lipids and proteins. So what is basic function of apoproteins? They can form a part of structure of the lipoprotein, like ApoB is the structural component of VLDL and chylomicrons. Okay, they are enzyme cofactors. For example, C2 for lipoprotein lipase A1 for lecithin. Okay, lecithin, we have discussed it in yesterday's class. And ApoA2, another example is ApoA2 and ApoC3 for lipoprotein lipase and ApoC1 for cholesterol ester transfer protein. Basically, they act as a ligand for interaction with lipoprotein receptors in tissues. Okay, for example, ApoB100 and ApoE for the LDL receptor, ApoA1 for the HDL receptor. Okay, so how they metabolize? Actually, in your syllabus, there is a topic of lipoprotein metabolism. So this is important. I think we should focus on this. Chylomicrons are found in chyle form only by the lymphatic system draining the intestine. They are responsible for the transport of all dietary lipids into the circulation. Okay, so basically chylomicrons are found in chyles, and if we where these chylomicrons and how synthesize and how they synthesize, so these chylomicrons basically synthesize in rough endoplasmic reticulum, and they contain apolipoprotein B48 type. And the synthesis of, as we see in earlier slide, that the synthesis of ApoB48 happens in intestine. And basically it results from RNA editing process. Um, what is RNA editing? Like we have a 100 amino acid mRNA having codons for 100 amino acid, and we want it only, I am just giving you an example, not exact, and we want only 48. So an editor RNA can cut the mRNA or cut the translation at, or truncate the translation of that mRNA at 48 amino acid. So this process would lead to the generation of only 48 amino acid containing containing protein, which will further help in the synthesis of chylomicron. So this is the RNA editing which actually acted at the mRNA level. How chylomicron synthesized? This is the codon. You can see these are all basically codon on DNA. They get transcribed, and through transcription, mRNA generated. Unedited mRNA reach to liver and intestine. In liver, whole translation and transcription. Whole mRNA get translated into apolipoprotein B100, which exactly contains 4,563 amino acids. Okay, and it functions in the transport of cholesterol in the blood. But what happens in the intestine? The same mRNA, it get edited. That is, after CAA, glutamine, it get stopped via stop codon, and translation stopped exactly half on midway. So there remains only 2,152 amino acid in this protein. So it is known as apolipoprotein B48. Basically, this is present in chylomicron, and its function is in absorption of lipids from the intestine. So the synthesis of ApoB48 basically happens due to the RNA editing process. If we move to the further part of this class, it is actually catabolism. In liver, a single ApoB gene is transcribed into the mRNA, as I already discussed it in this slide. So there is no need to discuss it again. I think if any one of you have any doubt, you can ask me anytime in the class or at the end of the class while I'm teaching. You can also ask any sort of question. Okay. So I'm not going too much detail on that process, because we have a lot of topics to study. In this flow chart, you can see this is the intestinal lumen. Here, these are blood vessels, lymph vessels leading to thoracic duct. Here in rough endoplasmic reticulum and in smooth endoplasmic reticulum, basically in RER protein synthesis occurs, and in intestine ApoB48 gets synthesized in RER and get transported via Golgi complex, and through exocytosis these ApoB48 comes into the lumen, and from here they basically enters into the lymph vessel leading to the thoracic duct. So this is overall the process of synthesis of chylomicron and transportation, and basically a beta lipoproteinemia, a rare genetic disease, may be caused due to the deficiency in synthesis of ApoB, and if its deficiency occur, basically lipid droplets accumulate in the intestine and liver due to non-formation of VLDL, which actually what VLDL does, it transports this ApoB from intestinal lumen to the other parts. Okay, so this is the clinical significance. And also sometimes it is seen that some apolipoproteins are having additional carbohydrate functional moiety. Okay, and from where it comes, actually, when after synthesis, when these lipoproteins or proteins transported to the Golgi complex, their carbohydrate residues are added, and these added carbohydrate residues in Golgi apparatus basically helps in the release of these lipoproteins through the process of reverse pinocytosis. Maybe you all are aware about the pinocytosis. It is the process known as cell drinking, okay, or liquid transportation happens via the process of pinocytosis. Transportation of chylomicrons basically lead to the passage of these chylomicrons into the lymphatic systems, and eventually they enter into the systemic circulations. How these chylomicrons get catabolized? Okay, so chylomicrons acted upon by the enzyme lipoprotein lipase, because they have specific protein moieties, so their specialized enzyme lipoprotein lipase is present to degrade chylomicrons. This actually causes loss of 90% of triacyl glycerols of chylomicron, loss of ApoC, which returns to the HDL, but not ApoE. This lipoprotein lipase degraded triacylglycerol and ApoC, but it does not act on ApoE. The resulting chylomicron remnant, which consists of ApoE, is about half the diameter of parent chylomicron and is relatively enriched in cholesterol and cholesterol esters because of the loss of triacylglycerol. So these chylomicron remnant basically taken up by the liver by receptor mediated endocytosis, and the cholesterol esters and triacylglycerols are hydrolyzed and metabolized. These uptake basically mediated by ApoE. So hepatic lipase has dual role. The first role is to act as a ligand to facilitate remnant uptake, and the other one is to hydrolyze remnant triacylglycerol and phospholipids. Basically, the products released, they are reutilized for the synthesis of VLDL. So the digestive products of chylomicrons, they are reutilized for the VLDL synthesis. This is the overall catabolism pathway. You can see in gut, basically this is the structure which we will discuss, which we were discussed earlier in our class. This contains both ApoC third, ApoE, ApoC third, ApoB 48, and ApoI triacylglycerols, and this actually metabolized via apolipoprotein lipase or lipoprotein lipase into the fatty acid, which further move towards the tissue for further degradation or deposition storage purpose, and the protein part, that is apo A1, A4, ApoC second and C third, get reutilized, recircled for the synthesis of HDL, and the remaining unmetabolized portion leftover is known as chylomicron remnant, which again are transported back to the liver, where they get entered via receptor mediated transport, and here actually they get recircled back and help in the synthesis of VLDL. So it is overall the fate of chylomicrons. Okay, similarly, I'm going to just tell you how VLDL get metabolized inside the body. So this is the structure of a common cell where RER actually synthesizes the protein part of VLDL, and smooth ER actually it is responsible for the incorporation of lipids, and combination of proteins and lipid moves to the Golgi complex where they actually transported back to the plasma membrane, and through plasma membrane via exocytosis or endosome mediated transport they can fuse to the neighbor nearby they fuse to the neighbor nearby endothelial cell of the blood sinusoid, and they entered into the blood sinusoid via this endocytosis mediated transport system. So VLDL are secreted into the space of Dicey. This is known as a space of Dicey, and then into the hepatic sinusoid through fenestrine in the endothelial lining. This zigzag structure is known as fenestrine in the endothelial lining. How it get catabolized, we can just see via a simple diagram. Here, as we already know that in VLDL, triglycerides, cholesterol ester, apolipoprotein B100, C3, C2, apolipoprotein E is present. It from liver, where liver is the site of synthesis of this VLDL. From liver, it get moved through blood vessel into the adipose tissue and muscle capillaries, where it get actually catabolized, and the fatty acids, free fatty acids released, which can be further utilized for the energy production or storage purpose, and the remnant part, which is the leftover part, is our cholesterol ester B100 and some triacylglycerol. This is known as the intermediate density lipoprotein. It moves back to the liver, and in the liver it get degraded again to cholesterol ester, triacylglycerol and ApoB. Every time when these lipid molecule enter into the any cell or any organ, this process happens via receptor mediated transportation, and these receptors are known as the low density lipoprotein receptors. Okay, and further these cholesterol ester and B100 is also entered through LDL receptor pathway into the hepatocyte cell, and here they helps in the synthesis of bile acid. Okay, this cholesterol, actually this cholesterol is utilized for the synthesis of bile acid, steroid hormones, and it also helps in the regulation, other regulatory processes, and membrane synthesis. So basically LDL binding to the receptor would lead to its endocytosis, and then which is followed by lysosomal hydrolysis and the release of cholesterol for the other synthetic purposes. This is the overall fate of VLDL in the body. How HDL synthesized in the body? We can learn this by seeing this pathway. This HDL basically it is synthesized into the liver from small intestine. Actually, the organs involved in HDL synthesis are liver, small intestine, and some part is also some part of kidney is also involved here. So in the liver, bile C and bile acids are produced, and basically HDL is present in two forms, HDL2 and HDL3. In HDL2, A1 cholesterol ester and phospholipid is present, and in HDL3, apart from this apolipoprotein 1, A1 cholesterol and cholesterol ester and phospholipid, LCAT is also present. Okay, this HDL2 enters into the liver through SRB1 receptor, and here it get its component like cholesterol ester and phospholipid is utilized for the production of bile C and bile acids, and it also converted into the HDL3, and this this would also synthesize back and enters into the other neighboring tissues, and this other neighboring tissues and helps in the synthesis of phospholipid bilayer. And it is also, this HDL is also come from small intestine, and from here it also get enters into the various tissues through receptor SRB1 and ABCA1, and in the tissue it get catabolized or move back to the other process, like it is also moved back to the kidney. Basically, in HDL helps in the transportation of lipid from liver to other organs. We discussed it in our earlier class that HDL basically helps in the transportation of lipid from liver to other tissues. And if HDL concentration decreases in our body, then liver get accumulated with this lipids, lipid part, and which causes fatty liver disorder. Okay, so to move or for proper transportation or distribution of lipids into the other part from the liver, HDL concentration is very important. And what LDL do? LDL basically transports the lipid from other organs to the liver, and it actually causes the accumulation of lipids into the liver. So this is the basic facts behind the HDL and LDL transportation. Okay, so now coming to the summary part, how this actually HDL, chylomicrons and LDL, they balance out each other. See, HDL, what HDL do? It transports excess cholesterol and fats from liver to cells. You can see these green arrows. Or it can transport these excess cholesterol back from cells to liver. And what VLDL and LDL do? They takes this excess fat and cholesterol for deposition from liver to the nearby cells. Okay, these star portion actually depicts the receptor mediated endocytosis, and they cannot transport without endocytosis. So receptor mediated endocytosis play important role. And what this chylomicrons do, they come from the intestine, that is enterocytes, and they basically enter into the liver, and here they involve in the various synthetic processes. So this is overall the balance between the HDL, LDL and VLDL chylomicron inside the liver cell. You can also see this like from intestine, this chylomicron comes and get metabolized, and it can the leftover, that is the chylomicron remnant, it can help in the synthesis of HDL, or HDL when it get metabolized, the leftover will be helpful for the synthesis of chylomicron. Similarly, in the liver, this VLDL synthesized inside the liver, and it moves outside the liver, and here it either convert into the VLDL remnant through catabolism, or it helps in the synthesis of HDL. So I already discussed a lot about fatty liver disorder in our yesterday's class. So I'm not going to not going much details on this, and let's just try to finish this. This is actually, our class will end in less than 1 minute, and we will continue this lecture, and if time permits, we will include another also. You people can rejoin this class using the same link, and if you have any query, you can ask in the subsequent lecture. Okay. So what is the basically transportation process involves inside the body while transporting LDL? So this VLDL, when it combines with ApoC and ApoE, HDL is also combined with ApoC and ApoE and ApoA, and it can also helps in the production of VLDL. And this VLDL basically, from where it comes from liver hepatocytes. Okay. And because VLDL is synthesized inside the liver. Okay. And after complete synthesis, it enters into the blood